So in this video, we are going to study about the primary growth in plants. That is the result in the formation of various tissues like xylem, phloem, parenchyma and so on. We will see how the primary growth in flowering plant takes place. This is an important topic from the chapter Anatomy of the Flowering Plant. Anatomy means to say the internal structure of the plant. That means epidermis, cortex, vascular tissues, parenchyma, collenchyma, xylem, phloem, sclerenchyma and so on. So how these tissues are formed? So they arise due to the process of cell division and differentiation during the primary growth. Now these cells, dividing cells, will result in the primary growth. You will see the example here. The seedling is growing where the roots are growing downwards and the shoot is growing upward, and this result in the increase in the length. And we are also seeing here that old leaves are produced at the bottom while the young leaves are present at the tip. This type of arrangement is called as acropetal arrangement. If we see in the detail, these cells which are continuously adding more and more cells to the overall plant growth, they are located at the apical region, that is the uppermost region. Here it is called as apical meristem. This will go on increasing the cells, and further these cells differentiate into various other tissues, and hence the length of the shoot will increase. Similarly, the length of the root also increases. So this is the primary growth, which is mostly due to the meristematic activity of the apical meristem. These meristem, the continuously dividing cells, so they go on dividing and increase the number of cells. This region is because uppermost region, that is, it is called as apical meristem. So in the case of shoot, they are situated at the uppermost region of the shoot, that is called the most of the distal region of the stem axis, and these are protected by the leaf primordia. While in the case of root, we will see they are also present at the apical region of the root, that is called the root tip, and this is also protected by the root cap. The activity of these meristematic cells, while in the case of shoot apical meristem, this increase in the length upward, while in the case of root, it is the increase in the length downward. The whole process involves cell division, then elongation, and then differentiation. So we will take the cell division firstly. So the cell division during the meristematic cells situated in the apical region, they go on dividing, and this will add the number of cells to the overall plant body. So addition of number of cells leading to increase in size is called as open growth. At this stage, we will see the three distinct regions are created. The outermost is called as protoderm or dermatogen. The inner is called ground meristem or the peribelum, while the innermost region is called as the procambium or plerome. These three meristematic tissues, they separately give rise to different primary permanent tissues. For example, the protoderm give rise to epidermis, the ground meristem tissues give rise to ground tissues like cortex, pith, pericycle, medullary rays, while the procambium or the plerome give rise to vascular tissue like the primary xylem, primary phloem. That is why these three regions are called as primary meristem. So from the apical meristem, we are now at the primary meristematic stage. Now the next stage called the elongation takes place due to the vacuolization and absorption of water. That means if you take the example of this meristematic cell having a dense cytoplasm and a central prominent nucleus, a small vacuole has been created at this stage, and this absorbs water, and this results in the expansion and the elongation of the cell. Next is the differentiation, whereby all the cells get differentiated into separate primary permanent tissues. The outermost region, that is called protoderm or dermatogen, differentiated to the protective covering called as epidermis, while in the case of root it is called as epiblema. The next tissue, that is called ground tissue or ground meristem, develop into the cortex, pith or medullary rays. The next tissue, so this cortex is differentiated into the outer cortex called as hypodermis. They sometimes contain the collenchymatous In the young stem, it is made up of collenchyma cell, and sometimes it contains the chloroplast, that is why it is green in color. The young shoot, they appear green due to the presence of the hypodermis collenchymatous cells that contain chloroplast. They carry out photosynthesis. While the inner region of the cortex is differentiated into the endodermis. Now, if we see this one, that is called procambium, which differentiate into vascular tissue, they cut off the cells toward the inner side, they form the xylem, primary xylem, and the outside they form the primary phloem. While some of the cells remain as such in the dividing stage, they are called as cambium. So in the most dicot and gymnosperm, the meristematic cells of the procambium remain undifferentiated, and it is present between the xylem and phloem. That is, it is not completely differentiated into primary xylem and primary phloem. Some of they remain in between, and they are called as cambium. While in the case of monocot, now we won't see that cambium also called as vascular cambium, fascicular vascular cambium. Also it is called as intrafascicular cambium. Now in case of the monocot and herbaceous dicot, this is particularly important, and vascular cryptogams, the procambium is completely differentiated into xylem and phloem. As a result, no cambium is left between the xylem and phloem. This is one major difference between the monocot and dicot plant, and particularly the herbaceous dicots. Now, if we divide this section into two parts to differentiate between the shoot side and the root side, we will see in the case of roots, the hypodermis region is absent. No collenchyma or mechanical tissues present in the roots. Collenchyma particularly is absent in the case of the hypodermis is absent in case of root side. And the root cortex contains the parenchymatous cells, which have a large intercellular space for the conduction of water, absorption of water. And also the endodermis region, that is the barrel shaped cells, this is the innermost form of the cortex. It is contain the Casparian strip. In the case of stem, it has a starch sheath. Pith is generally in root, dicot root particularly, it is absent. And the xylem, if you see, most of the region is occupied by the xylem, where small xylem vessels are present toward the outer side, that is called protoxylem is toward the outside. This condition is called as exarch. The phloem is alternate with the xylem, and this type of arrangement is called as the radial arrangement. Root also, the root epidermis or epiblema also form the root hair. The epidermal cells get modified in unicellular hair for absorption of water. While in the case of the stem, we will see unicellular to multicell projection from the epidermal cells. Some are glandular. These are called as trichomes. The stem of the dicot, they have a thin cutaneous layer that is called waxy coating on the surface. This is thin cuticle layer. While in the case of root, the cuticle is absent. The another thing difference between the root and stem is that in the case of root, lateral root arises from the pericycle cells from the inside, that is called as endogenous formation. While in the case of stem, axillary buds arises due to the apical meristem, and they are formed from the apical region, that is called, that their formation is called as exogenous, and they may form a shoot, a branches, or a flower. So in total, we will see that apical meristem give rise to the primary meristem, that include the protoderm, ground meristem, and the procambium, that later forms the primary permanent tissue, epidermal tissues, ground tissues, and the vascular tissues. Together they are called as the primary permanent tissues.